Well, good afternoon, or almost evening. Thanks for the invitation. It's nice to be back. I've been here in 1968, 54 years ago, pretty much to the day or to the month, when most of you were not yet born, including my own children, not even in the planning stage. So, well, in the meantime, we've done something, and I'll try to tell you a little bit about it. These are the guys, some of the people that have contributed to that work, including, I have to say, conceptual discussions with Tony Dickinson, Chris Harris, an economist in Cambridge, and Charlie Plott, an economist in Caltech, where I've been visiting in the past years regularly. Of course, you know that. This is some of the people that have contributed to the work I'm talking about today. Maybe that was 3-4 years ago. Some people have left, other people have joined that are not in the picture. Bill Stauffer and Armin Lack are the most responsible for the second part of what I'm talking about with their enthusiasm. You can see that with Armin. Bill is more relaxed, but he's very enthusiastic, but he's also enjoying his work right here. Okay, that's in front of the department. I'll take you to a walk through Cambridge. This is my currently favorite pub, the Maypole. It's not a great pub, but it has a great atmosphere. And they have, of course, it helps us to understand what we're doing. They have beer, and you can choose between beers. A proper job. I tried to find today a similar picture from the Krakow pubs, and I could not find it. I apologize, I understand. Bringing beer to Krakow is a bit ridiculous, but that's the way it is. And the other is Adnams. That is brewed actually close to Cambridge on the east coast towards the North Sea. So I'm an IPA guy, and I like these modern IPAs more than these standard beers. And so before coming to Cambridge, I didn't know any of these here. I didn't know these one either. That's, by the way, another Cambridge brewery. Anyway, I like it, and I tried it, and I wanted to have it again. Right? I tried that one. This is pretty good, but not if you have that one. You don't do that anymore. I don't try that anymore. Maybe occasionally, but very rarely. So I have developed a preference for proper job as compared to the ghost ship, and as compared to these two beers as well, I have to say. So I've learned by repeated experience that this sign here, proper job, indicates a beer that I like. And that sign indicates a beer that I would probably not take if this proper job is available, right? So I'm going now, this is my whole talk, from learning to decision making, okay? And the idea is that learning updates the value of this stimulus, and then when I try this repeatedly, I will hopefully continue to like it, and then there will be no much change in my liking, so the reward value will stay the same. I won't have a prediction error, but I need to keep trying here, otherwise this stimulus will go into extinction. This one is the same. I will also update it, but it will not change my lower value appreciation, right? So the idea is that this reinforcement learning serves to update the values of choice options, right? This is one option, that's the other option. I'm trying to introduce some terminology that we need. And of course I have a choice, right? The options have a value, the choice doesn't have a value. A recent referee told us that we're all wrong, choices have value. I'm sorry, that's not true. The choices are only made between two options, and these options have values. And I try to say that I will choose, of course, what has the higher value, or the other way around. When you see what I'm choosing, proper job compared to ghost ship, you will infer that proper job has a higher value for me than ghost ship. Okay? Learning all started 100 years ago, the scientific investigation of learning, with Pavlov. He had this doggy that likes sausage, just like I do, and then the dog salivates when he looks at the sausage, exactly like I do as well. Okay, the unconditioned response. I cannot do anything against it. I will salivate when I see a nice Polish sausage, kielbasa or something. But then somebody rings a bell at the same time, or maybe a couple seconds before the sausage appears in front of me. And then if that happens repeatedly, I will start salivating to that bell, right? So the bell has suddenly become something like a substitute for that sausage. It elicits a very similar response as the sausage, but before the sausage appears, okay? So we formalize this. We have our sausage here. We have our bell that rings, or visual stimulus that we use in monkeys, far better controllable. And then if you do this repeatedly, some of the behavioral responses to the reward, like the salivation, will be copied onto the stimulus. I will still salivate to the reward, but I will also salivate to the stimulus, because the stimulus tells me that I'm going to have a sausage. The prediction of reward, the prediction of sausage, I should say here. Okay, that's the whole idea of Pavlovian learning. Very simple if you bring it down to this very simple graph. And also, I do not need to do anything. I do not need to eat that sausage. I will salivate because I'm me, or it's the dog that will salivate to the sausage. Whether I grab the sausage or not doesn't matter. Okay, that's the point. Not operant condition. Operant condition, you need to do a movement in order to get that sausage. But the stimulus still in operant conditioning is Pavlovian conditioned, because the stimulus predicts the sausage, and then in operant conditioning I will do a movement to get that sausage. Okay? Right, so let's go into the brain. Dopamine neurons, you may have seen this, that's a human brain actually here. Dopamine neurons here, small neurons. We record, that is a classic thing, we still do that. With the electrode, the action potential from extracellular positions, okay, no patch clamp, nothing, in awake animals, through an amplifier, and we pass this through a threshold discriminator to produce computer-compatible TTL pulses, and we plot these pulses over time, horizontal. Time goes here, 0, 1, 2 seconds. The monkey sits in a chair in the laboratory, and he does a movement whenever he wants it by releasing a resting key, a touch-sensitive key that doesn't move, and then goes into this food box that is covered, so he cannot see into the box, but he can reach underneath into that box. And he does it whenever he wants it. If he goes too fast, we don't give food. If he goes too slow, we increase the food. It takes maybe 3 months to train an animal to do that in a proper way, but it's doable. And we look at the muscle activity to see whether the animal has isometric contractions or not. And this is wonderful in a monkey. They're wonderful animals. You can control their behavior. If they contract the muscles, you just don't present food in there, or you make sure that he will not get anything. Maybe not in this task, but in other tasks. So we have control over the monkey's muscle activity and movements. And what we see is that at the time of the movement onset, that means the release of the resting key, the number of action potentials per unit of time increases. This is a burst of action potentials. That's one trial. There's 2 trials, maybe 15, 20 trials, and that's the sum of all this. And what we see is a higher rate of action potentials. We call that a neuronal signal, right? This is background activity, something happens, and that happens is a neuronal signal. Okay, there's a little inhibition afterwards, that's not important at the moment. All right, so is this because the animal starts to move, or is that because the animal is getting food or anticipating food, whatever. So we plot this a little closer. This is the animal touching the piece of apple inside this food box here. And that is held by a touch-sensitive wire. You just put a wire into a field effect transistor amplifier and pass the signal through a Schmidt trigger, and you get a TTL signal. And you can plot the signal here. And so this is before the animal touches, and this is after the animal touches, or while and after the animal touches. And you see that the dopamine response is very consistent here, not always consistent, but pretty consistent, when the animal touches this piece of food, a piece of apple, 1/100th of an apple. When the animal touches the bare wire, he still touches, he still has done the movement, he still has somatosensory stimulation. We still trigger this TTL signal here, but there's no food in there. And the animal will move around, actually search for the food, he won't get the food, and there's no response. Okay, so this is not related to the movement, it's not related to the somatosensory touch, which was an issue at the time. It is related to simply that there is a piece of apple. Okay, so a piece of apple. Why are monkey neurons interested in pieces of apple? That's a bit odd, so you want to do a proper test. Okay, what you do is you take a liquid, a drop of liquid that comes through a spout at the mouth of the animal. And you can open a electromagnetic solenoid valve from the computer for 50 milliseconds, 30 milliseconds, whatever. How long you open it determines how much liquid will flow, as 1 or 2 drops at the mouth of the animal. That's what happens here. And you see repeatedly the dopamine response to that. Of course, we do that, we deliver the food at a time that the monkey doesn't know. We have a little button on the computer keyboard, the F for free reward, and we touch that, animal doesn't hear nothing, and suddenly the liquid comes, okay? Then we give a stimulus, now a visual stimulus, something like I showed before, in anticipation of that reward. The animal does not need to do anything, or it can do a movement. It doesn't matter whether we have a Pavlovian or an operant task. What we see, like 1.5 or 2 seconds later, there's a response to the stimulus, but not later, not to the reward. Oh, that's very funny. So we have a beautiful reward signal that is general for food and for liquid reward. And when you test it properly, the response is gone. Okay, that's a problem. So we have a reward signal that is not a reward signal. What do? You play around. You sit there and you tell each other jokes, because we were normally 2 people in the laboratory. When we were telling jokes, the monkey didn't perform the task well, made mistakes. And when the animal makes a mistake, it doesn't get a reward, right? So, sorry, I forgot the theory here behind. This transfer from the reward to the CS reminds us of the Pavlovian transfer here, right? So the neurons are Pavlovian conditioned, okay. All right, so we still show the stimulus, but the monkey doesn't care. The monkey doesn't pay attention because he hears us laughing and he thinks we're not serious, so he's not serious. And what you get is, at the time at which the reward should come, even if the monkey doesn't pay much attention, the neurons shut off. And so they're just silent at the time at which the reward should have occurred. We can also delay the reward artificially. Once we discover this, we play around, of course. We can delay the reward, and the same thing happens at the original time of the reward. When it does not occur, we have a depression, and at the new time, we have an excitation, just like this excitation, right? So that is not very complicated. We see there's an element of unpredictability in there, and that is formally referred to when you go to the standard textbooks on animal conditioning as a reward prediction error by Rescorla and Wagner. So the idea is that this is the equation. The reward prediction error is the reward received, the so-called lambda, minus the prediction. Well, we have to say this is expressed in milliliters of juice. So the value, the prediction, is a prediction of milliliters of juice, and the reception is also a certain amount of milliliters of juice. And here we see there's no prediction. This reward occurs irregularly. We press this free reward button. Here the reward occurs exactly as predicted, not only in the amount, but also at the time. That's important, right? And here, the reward does not occur, although it is predicted. So you take that equation, reward received is 0 minus reward prediction, that is negative. The reward prediction error is negative here, the reward prediction error is positive here, and there's no reward prediction error, okay? Then, as I mentioned, this goes very well with the Koller-Wagner learning model. This based on the unpredictability from K-means blocking task. The lambda minus v is the crucial term that determines the learning. You have a learning constant that determines the speed, and you take the current prediction, you add the prediction error weighted by this learning constant, and you get the next prediction. And when you plot that over repeated trials, you get this gradually asymptoting function. Okay, and that's what the dopamine neurons are doing. All right, and then when we take our control theory block diagram, we can specify it that the prediction generates the behavior. So learning is defined as a change in behavior. We had discussed that yesterday. And of course, as a prediction influences the behavior, when you change the prediction, you influence learning. That means a change in prediction means learning. Okay? You get the reward, you compute the reward prediction error, and then you use this to update the prediction. Okay? This is an error-driven learning system, but it is not an error-correcting system because there is no target state that needs to be reached and from which one computes the difference to the outcome. Right? The reward prediction error will increase the prediction, the positive prediction error, and the negative prediction error will decrease the prediction. Okay? But there's no bound to the positive prediction, right? So it's not an error-correcting mechanism. It's an error-driven mechanism because we're talking of an error, but we're not correcting anything. The prediction is not a state that needs to be maintained, but the prediction can increase. Okay, right. All right. So this was the situation we had seen. In fact, before we saw this reward prediction error response, that the neurons are active doing movements. There was no prediction error, and we had a very complicated task, as people were doing in the 1970s and 80s, but everything was predicted. And what we saw was some movement activity when the animal made a big reaching movement. When the animal makes a small movement or an eye movement, there's no dopamine activity. Okay? As soon as we controlled the task much better, and introduced prediction errors inadvertently because we had reduced the task and then the reward was unpredicted, we saw this funny dopamine response. And then we characterized it as reward prediction error. So with a positive prediction error, the response is strong. With the smaller positive prediction error, the response is mild. And with the negative prediction error, we have a depression of activity. Sorry, that happens at the time of the reward, but we also saw that at the time of the stimulus. We'll come to that in a minute. Okay? But then what we saw later was that these responses are preceded by a sharp earlier response that is related to attention. We'll come to that also in a minute. And then we go back to these movement-related activities that occur in between the stimulus and the reward, or before the stimulus, or before after the reward, or at any time. This is what people see now, and there were half a dozen Nature papers on that. In mice, when they test dopamine neurons, they suddenly see either no prediction error, when everything is predicted, although they don't always realize that, or what they see most usually is the prediction error plus some movement activity. And there were half a dozen papers on movement activity in dopamine neurons as if it were a surprising fact. And of course, there is the tonic activity of dopamine neurons, this tonic background activity that's necessary to prevent Parkinson's disease. So what you now have is a super complicated signal, right? If you read the papers now, people are confused because they see all this blue stuff here, and where we usually talk only about this red and maybe this orange or yellow, brownish stuff, okay? So the idea was that dopamine neurons do prediction error, and if they do the prediction error during this period, which is fully learned, then the prediction error is really useless. But that was under the impression that one brain system has only one function. What we see is, that is the old idea. The orbitofrontal cortex does that. The amygdala does that. The dopamine neurons do reward, right? Unfortunately, that's not true. We think these are independent, separate functions, like movement function and the reward function. And they're not, as people like to publish, categorically separated between the medial dopamine neurons, the VTA, and the substantia nigra pars compacta. It's not true. There is a gradual difference where the VTA neurons respond better to rewards and the substantia nigra pars compacta neurons less to rewards. Okay? Right, but to make things simple, and in particular to take the most interesting element out of these dopamine neurons is the reward prediction error. So we'll only look from now on onto this phasic response here. Okay? What I forgot is this yellowish response here, and we'll quickly talk about that, but then we focus on this. So a reward, when it comes, for example, to the lip, has a somatosensory stimulus property. It touches the lip, right? That's not a reward. That's simply like my finger touching my mouth at the moment. But then, of course, I myself and my neurons, including my dopamine neurons, distinguish between a drop of saline and a drop of apple juice, right? Even though it may be somatosensory, the identical stimulus, at least in the beginning, until it hits my taste receptors. And then I distinguish between the different liquids, and then I say, well, I like grape juice, but I hate saline, right? Then it becomes a value signal, okay? So we have at least two components: the sensory impact that is partly related to attention, and the positive reward value, okay? When we give a stimulus in random alternation with a neutral stimulus in random alternation with a reward predicting stimulus, we often see these responses here. But when we then take another stimulus and that stimulus predicts a reward, the response is much stronger. And so we think that we can distinguish here a sensory component. The visual stimulus comes, and that's now this reward predicting stimulus. And then later the value. The no reward has no value, and the reward itself has the positive value, okay? And this is not the only system in the brain that does it. After we realized what was happening, and it took us maybe 20 years to figure that out, through another experiment that I don't have the time to show, we realized then that in fact, this is not the only brain system. You can find that in the frontal eye fields, for example, which respond in a task. This is almost 30 years ago now, or 25 years, in a task in which there is a target stimulus or a distractor stimulus, and the animal has to react to the target and not to the distractor, that when the stimulus occurs, there's the same activity for the two stimuli, but then it deviates later into the target-related activity dies down for the distractor. And I can find a list of 20 studies that show very similar things throughout the brain, in the visual cortex, in the amygdala, in the striatum, wherever you like, orbitofrontal cortex, okay? Then came the question, well, we get some active excitations of dopamine when we present a drop of saline, right? So, of course, saline is a punisher. It has, again, sensory input, and it has an aversive value, the negative value, all right? So, this is what people say. They do a stimulus, an aversive stimulus, a stimulus that predicts 1 millimolar concentration, a drop of a bitter liquid. It's called denatonium. It's a apparently terribly aversive liquid, right? Nice response, excitation. So, is that a reward signal? That can't be, because this is a bad stimulus. Okay, so people say dopamine neurons respond to aversive stimuli. Well, they do. Look, we increase the concentration of that aversive liquid. We make it even more aversive, and the response, the green response, does not go up. The green response actually goes down. So our interpretation from this and many other experiments is that the aversive liquid has negative value. The negative value cuts this initial excitation, produces an inhibition that cuts this initial excitation. So there were a number of studies that showed aversive excitations in dopamine neurons. And when we go and look at the controls they have done, they have failed these controls. There were only a single paper from Uchida's laboratory. He also did not do that control, but he used a liquid, a small amount of liquid that drove the excitation of a few dopamine neurons in the posterior lateral substantia nigra, and also the fiber recordings in the posterior striatum that actually showed an excitation. But when you used another liquid, the excitation went away. So it's not the physical response that he saw. He probably saw an aversive response for a particular aversive liquid. So what I'm trying to say is most dopamine neurons actually code reward prediction error, not an aversive prediction error. Oh, and these Uchida responses did not code a prediction error either. Okay? Good. Having done that, we play around a little bit, going closer to what I want to say. An increase in reward amount increases the dopamine response. 0.05, 0.15, and 0.5 milliliters increases dopamine responses. We know that reward value is determined not only by the reward amount, but also by the reward probability. How much and how often we get reward. Okay, so we can do an experiment. We can use our scheme of the reward influencing the value of the stimulus. We can use different probabilities. So in 10 trials, the reward will occur in 8 or in 5 or in 2 trials, right? This is a high probability, medium, and low probability, right? So the animal gets a stimulus and then rewards come, not in every trial, right? So this frequency, this is the frequentist approach to probability theory. This frequency translates into a probability and the probability approaches this frequency if you use enough trials, okay? So we are transforming the experience reward into a predictive stimulus, a predictive probability. And here is the experiment. We use a familiar stimulus that always predicts reward at a probability of 0.5, and we use one or another or a third novel stimulus, and the animal has a choice between one of these stimuli and that stimulus, right? And these 3 stimuli will predict the reward with 3 different probabilities. So what does the animal do when we give him new stimuli here? This is the same stimulus always, but we change the stimuli every so often, like every day or so. What the animal does is, the monkeys are very nice, they want to see the novel things, they try out the novel stimuli, 100%. And with increasing trials, their choice of the novel stimulus is high for the stimulus that ultimately will predict the reward in 3/4 of trials, and very low for the stimulus that predicts the reward in only 1/4 of the trials, and the other is in between, right? So from the initial novelty behavior reaction to all novel stimuli, the percentage of choosing the novel stimulus deviates quite well between the different probabilities, right? So we can say that this stimulus here has a higher value than that stimulus, okay? Right, so then we look at the dopamine signal doing learning, and we see an initial peak, like this initial response, and that decays gradually with the trials, irrespective of which stimulus is shown, right? So this would be our initial salience novelty response, generating attention. And then the later stimulus component, that's the response to these stimuli individually. We see that these responses are initially non-discriminant and later discriminate according to these 3 reward probabilities, okay? So we in fact see that the dopamine neurons draw a value signal out of probabilities, which makes sense, and we see that this is a predictive signal, okay? But then there's more to that. When we played around with these tasks, and that's this paper 1993 that Dieter mentioned, we not only give a reward, but we also give a stimulus before the reward, what I showed initially, And we may even give another stimulus in front of that stimulus, in front of the reward. And what we see is that the neurons respond to the first unpredicted event, the reward, or these conditioned stimuli, which are higher order rewards, and not to the fully predicted subsequent stimuli and rewards. Okay? So, we are coming to another learning model. We're going from the simple Rescorla-Wagner learning model to temporal difference learning model, in which case, which is based on the Bellman equation, and the Bellman equation tries to maximize the value of the so-called value function by adding the current prediction and the discounted sum of the future predictions, of the future rewards. So, well, this would be the reward at that point here, the lambda, and that's the prediction, whatever the nomenclature is. And it subtracts the current active prediction, right? So when the animal would do a choice between these four rewards at that point, at that point, at that point, the animal would choose, of course, the better reward. And that would maximize this prediction error, right? So this is the idea. And so here is the idea. This is the Bellman equation, the maximization of the value function. This is the temporal difference learning model from Sutton and Bartow, in which we have what I just explained, the Bellman equation minus the current prediction. This is the prediction error now. And then, of course, that goes back onto Rescorla-Wagner, in which he has this term of the reward simplified in a single reward. We're not talking of CSs, we're talking of simple rewards minus the current prediction. And that, of course, is a formalization of Pavlov. So we are back to Pavlov in this case. Everything starts with Pavlov, and we have this nice temporal difference learning model with the that tries to solve the problem of the Bellman equation, right? So somebody asked yesterday, I'm sorry, I could not understand the question, what the dopamine neurons are doing, how they, and I forgot the question exactly. And the answer was they're maximizing the value function. So I'm trying to show this here. You can actually, this experiment has not been done. You can actually make a formal experiment. You would normally, when you have a choice, let's say I make it complicated, between four options, you choose the best option. Size is the amount of reward, right? You choose the best option, and the next trial, you choose again the best option, and then the next, and the next, and the best. And so you're maximizing, by these choices, you're maximizing your immediate and your future, your reward. But that's not exactly what the Bellman equation is about. It's true, but it's not all. What it is, what the Bellman equation says is you want to maximize the total reward value, right? This is the total, this is the current reward plus the discounted sum of all future rewards. So you have a choice here. You would, of course, choose the four. There's no alternative. But now there is a problem. You can choose this one. This is very nice, much better than this. But you see later, you're not going to get a lot. Right? So this is when you give up studying and you become a banker, you make a lot of money, but you're not going to increase it very much afterwards. Or maybe not a banker, but a plumber or something. The other possibility is to go and study. You won't be very rich, but later you're going to be a banker. You're going to be really rich. So, okay, here's the thing. If you maximize in every step, you're okay. But that's not what happens. What you do is, you may even go for a lower reward because the later reward is larger. This is maximizing the value function. And I postulate, and that experiment needs to be done, that the dopamine neurons would actually code this when you go through this here and not when you go through this here. That means you can impose on the animal this sequence, and you can impose on the animal that sequence and have different stimuli here, although they may all be the same reward, and you want to see whether the dopamine neurons respond higher to that stimulus for that sequence as compared when that stimulus predicts that sequence. Okay? So this is what we think dopamine neurons are doing, maximizing the value function. All right? And yes, of course, and you need temporal discounting, so I will quickly go through there because time is advancing faster than I do. We present 4 different stimuli in random alternation, and each stimulus predicts a reward after a specific delay, 2, 4, 8, or 16 seconds. And the animal has a choice between one of these delayed stimuli, and always the 2-second stimulus. This is constant, and this is one of these 3 delayed stimuli. And then we do a psychophysical titration to get the indifference point. How much reward do I need to give the animal that it chooses each of these 2 options 50%? And what we see is that we have to give much less reward when the animal chooses between the 16-second stimulus and that early stimulus compared to the 4-second stimulus, right? And then we plot this, and we get a discounting function that's hyperbolic, slightly better fit than exponential discounting. The physical value does not change, but the subjective value goes down. This is the value in the common currency of this 2-second reward, right? Okay, and then when you look at the dopamine responses to these stimuli that predict these rewards outside of a choice, we see that they have a very similar temporal discounting, also hyperbolic. Okay. So temporal discounting works on dopamine neurons, which is a requirement for this Bellman equation to and the temporal difference learning to function. Okay, what I said is biology. When then we go to the computer, and Demis Hassabis group had probably half a dozen papers on the game of Go and shogi and also chess now, in which a reinforcement learning model with a deep learning algorithm, with a lot of deep learning algorithms, learns to play Go much better, or better, than the best human players, right? So the standard reinforcement learning standard reinforcement learning together with the deep learning is better than the human player at the end, and is even better if there is a human supervisor that produces quicker learning, but then does not reach a high enough asymptote, right? So this is the computer implementation of a reinforcement algorithm, temporal difference learning, showing the power of this algorithm, but of course together with deep learning. Okay. But this is a computer. And the Bellman equation runs with numbers, okay? The implicit assumption in the Bellman equation is that these numbers refer to the value that's important to the individual decision maker. The Bellman equation is a reward maximizing description or equation. And I don't need 10 steaks in a day, although that is more than 5 steaks. I don't even need 5 steaks. The Bellman equation assumes tacitly that this value that's been maximized is the value that's good for me, not the physical amount. For a computer programmer, it doesn't matter. That's a number, right? In biology, that's the whole point. We need to show if this dopamine signal has any real meaning or any direct biological meaning, that the neurons are coding subjective value and not the objective value. This is the rest of my lecture. It will take a while. So the sensory receptors translate the impact of external signal into neuronal signals. That means we learn that the value of the deflection, the simple number of the deflection, translates into action potentials, and that's crucial. Now we can see how we can get our action potentials up. There are no specific receptors for reward, right? We have only somatosensor, visual, and so forth receptors, but no receptor that tells me exactly what is good for me, right? Oh, here you go. Now you're going to have a barbecue, right? And you're going to have a steak here for everybody, Daniel, and then you may have a second steak for everybody, but you can be sure, no problem. They won't eat 3 steaks. Nobody will eat 3 steaks, right? Even though 3 is more than 2, and 2 is more than 1. Right? You don't, it's the same receptor stimulation, the same steak. You don't do that. You just eat 1 steak and maybe, if you're really hungry, a second one. But the next steak is no longer a reward. So rewards don't exist. They don't exist in terms of physical entities that stimulate your receptors. They're made up by the brain, because the brain determines what you need. The brain determines whether I'm eating a second or a third steak, right? And hopefully, I won't eat the third steak. And so are the neural reward signals, because the brain works with reward signals. So if the brain makes up stuff like reward, it's the neural signals, the neurons that make up that stuff. That's what I'm trying to talk about. Okay? So we need to formalize a little bit what I'm going to say. I don't know how much time I have left. Yeah, minus 1 minute. You have the animal choose between two options. They're separable. They're mutually exclusive. You can only get one or the other. What the animal sees is what he can choose. There's nothing else. They appear simultaneously, which is important to not have some transient adaptation, that nothing intervening, and the cost remains constant. And with repeated trials, which you need for statistics in neurophysiology, the choices become stochastic. You do again and again, and you may have a little noise on top of your decision mechanism, and you change your choice sometimes. You never choose 100 to 0%, but you choose 70 to 30 or something. Okay? So we infer the value from the probability of choice. That's an important thing. We cannot measure the value in behavior, maybe in neurons, but not in behavior. We have to observe the behavior, and we say that the choice shows the value. When I choose A more frequently than B, then it shows that then we say A has a higher value than B. And then we can see if we can find a signal that follows that value. So the probability of choice is our substitute for value, okay? So here we can do that. The animal chooses between two stimuli that predict 2 seconds later a piece of banana or drop of liquid. And the animal does an eye move in this case to this or to the stimulus, and we've realized that the animal chooses each one 50%. The animal's choice is different between the two. So we say the two have the same value. Okay? Then the animal chooses between the same drop of juice and a larger drop of juice, and the animal hopefully prefers that drop of juice. So we say this option is preferred over that option. That's the preference symbol. Okay? And the same thing here, big drop of juice and 3 pieces of banana. Animal prefers the 3 pieces of banana, and we continue. And here's a large drop of juice over 3 pieces of banana. The animal still prefers that. Okay, so we have a sequence of a ranking of the values. This value is highest, and this value is lowest. Okay, and the neurons do the same thing. So if we analyze this thing, the yellow line is this one here. That's the highest value. The dotted line is this here. That's the lowest value. And this one, the green line, is the intermediate value. So there's basically no response to that intermediate value. There's a positive response to this top value and a negative response to that negative value. Of course, this is a stimulus that has undergone reinforcement learning, and at the time of the stimulus, the current prediction is somewhere in between here, in the middle. I don't exactly know where. Somewhere in between. And if if the stimulus comes for this reward, then that produces a positive prediction error. When the stimulus comes for that little piece of banana, that produces a negative prediction error, right? So we got a hierarchy of prediction error responses from negative to positive that follows exactly the animal's preference. That means it follows the animal's value. Well, that's the subjective value, okay? Because we see that from the preferences. All right? Can the animals actually distinguish good from bad rewards? So we are using a option that is a fixed reward that's small, and another option that has a small and a slightly larger reward. Each one of these rewards occur with 50% probability. Always one of the two will occur, but never both. Okay? The animal can choose this or can choose this, right? Well, if you look at it, the red option is probably better than the blue option because it has 50% more, right? It's like when I offer you 10 euros or 10 euros 50% of the time and 20 euros at the other 50%, you choose the second option, right? And that's what the animal understands, okay? Now, this could, of course, be the animal is simply risk-seeking. The animal doesn't understand these stimuli. The animal just understands this is riskier, something fishy about it, and that's not fishy. So we need to check, have a control for that. And the control is that we now use the same stimuli here, but we elevate that safe stimulus to higher. The animal will always get this blue reward when it's the same juice, when it chooses this option here. And if the animal were risk-seeking, it should still prefer that option. And if the animal knows what it's doing, it should prefer the blue option. And as you can imagine, why I'm presenting this, the animals know what they do. This is the stochastic choice, right? Oh, okay. And the neurons do the same thing. Dopamine responds to these stimuli. Now to have them visually more similar, we use both stimuli with 2 bars, but this stimulus, the red one, is better because the lower bar is higher, right? So the animal will, on average, get more reward here than here, and that draws a stronger response. That's also referred to as forced or stochastic dominance. The subjective reward with the sequence of preferences and all that, and temporal discounting is all fine, but it's, for me personally, too unconstrained. There is a mathematical definition of subjective value in economics, and that's called utility. Very simple. And that was defined by Daniel Bernoulli, who said that the utility gradually declines the richer you are, and was formalized in the von Neumann-Morgenstern axioms. Okay? And so we can try to get a utility function from the monkeys. We do our choices again between the safe and the risky reward. We follow a particular procedure that I don't have the time to explain, the fractal procedure in which the animal chooses between different sets of options and that are systematically changed. And this is the utility function that we get. It's not a psychophysical function. It's initially convex, linear, and then concave. Okay? And then we plot that utility function and plot the amount of reward that we give the animal, the free reward, and that the response increases in this nonlinear function. The black responses are the neuronal responses to these different amounts of reward, and surely these black responses do not follow the line of the physical value, but are more nonlinear, like this utility. And then we can do this a bit better. We can look at the specific prediction error response, at a well-controlled prediction error response, and we'll just take again the utility as a function of the physical juice reward. We plot our utility function. We take a gamble, again 50% top, 50% this bottom reward. The reward prediction error is shown here, that's in physical amounts. We take, and the utility prediction error is read over on the y-axis. We take the same gamble, the same distance here, but we plot it at the lower mean value, and we compute the utility prediction error, and that's slightly bigger than that one. And then we take a third gamble at the very flat part of the utility function, and the same reward prediction error, again, produce a much smaller prediction error in utility. So, with the identical physical reward prediction error, we're getting 3 different prediction errors for utility that depend on the steepness, the local steepness of the utility function. And here is the dopamine signal, as I explained, with a positive prediction, with the reward that's bigger than the mean, we get a positive reward prediction error and an excitation, and when it's smaller than the mean, we get an inhibition, but that is a little bit more complicated to measure. This one is quite easy. And when we look at this, we see that the positive reward prediction error response is higher at the center of the utility function than in the periphery, although the physical reward prediction error is the same. So the prediction error follows the utility function and not the physical amount. And you can rewrite the Röskala-Wagner learning rule or the temporal difference rule in terms of utility prediction error. That means the reward prediction error is a term, but it has a metric, and for dopamine neurons that metric is utility and not milliliters of juice. Okay? Well, I don't know. How much time do I have? I should probably, 5 minutes? Okay. One more experiment, and then we should probably cut it short. We can now see what dopamine neurons do with risk. By risk, we don't mean the probability of a damage, but we mean the variability. So this is a top, a very high, and a very low reward. That has a large variability, and this reward, this option has a certain reward and a little smaller reward. They have the same mean. We say this one is more risky than this one because you exactly don't know what you're getting here, whether you're getting really good reward or really bad reward, whereas here you get pretty much a decent reward, right? So this is riskier than this here. In economics, the risk is always expressed like that. This is in fact the variance of a probability distribution. So we can now plot these two gambles onto our utility function. The mean is the same. The outcomes are the two extremes, or these 2 points. And we see that when we simply take pencil and paper, we plot these gambles, we see that the utility, or the expected utility, that's the mean utility between these two utilities, is higher for the red gamble than for the blue gamble. So what I'm trying to say is the red gamble has a higher expected utility than the blue gamble. Okay? If that is true, if a utility function predicts choices, the animal should choose the red gamble more frequently than the blue gamble. That's the whole idea of a mathematical function. You take some measures, you fit a function, from the function you can make predictions. In this case, we predict that the animal chooses the red gamble more frequently than the blue gamble. And as you can imagine, how I present it, that's indeed the case. Not 100 to 0, but 65 to 35 or so, okay? What do the dopamine neurons do? They show the same thing. To the gamble, when you present the monkey with this gamble, the response is higher compared to presenting the animal with this gamble. Right? This is a value response. I've shown before that the neurons follow the reward value. This is not a risk signal. What it is that the neural response is enhanced. The value response is enhanced by the risk, because the riskier reward has a higher value for the animal than the non-risky reward, because the animal chooses this riskier reward more frequently than the other one. Okay? So again, a subjective value. This is a kind of an out-of-sample test for the utility coding. Again, we can, from the utility function, predict the choice, and from the choice we see that the neurons are in fact coding this value that we can infer from the animal's choice. All right. Well, maybe this one thing here. Okay, our scheme. You're familiar, probably, some of you, of the electrical self-stimulation that's now known for 70 years. And for 40 years, it's known from Roy Wise's work that 50% of these self-stimulation spots in the brain are linked to the dopamine neurons. That means the animal presses a lever in order to get an electrical shock in his dopamine neurons. And it will do it again and again and again. This electrical shock, if it were really dopamine neurons, would replicate a positive reward prediction error. Right? And that's an excitation. The electrical shock produces an excitation in the neuron, and that excitation is very similar to the response to a positive reward prediction error. Right? So the electrical elicitation, as we now know it after this time, the electrical elicitation of this behavior indicates that the animals are seeking dopamine excitation. And there have been a number of studies on optogenetics to confirm this, and we have done this in the monkey, and we have infected the monkey with channelrhodopsin, not terribly efficient, but pretty good, pretty specific, 95%. And what we have done, as you can imagine, is a choice test in which the animal chooses between two options: a blue option, a blue stimulus, 2 seconds after which the animal receives a drop of liquid plus an optogenetic stimulation, channelrhodopsin stimulation. after the red option, the animal receives the same drop of liquid, but no optogenetic stimulation. And when we look at the choices, we see that initially the animal is pretty much indifferent. It doesn't know which one of these stimuli are good, but after a while, let's take the blue curve here, after a while, the animal figures out that in fact the one option, and that's the stimulated option, that's this one here, is better, and the animal chooses this more frequently than the other option. And the red curve shows the same thing with electrical stimulation. Now that highlights that in this case, the electrical stimulation was far more efficient, probably because the channel rhodopsin stimulation opens the channel that depolarizes the neuron, that then only later depolarizes the voltage-dependent sodium channels, whereas the electrical stimulation directly goes on the voltage-dependent. It goes directly on the membrane potential, which opens the voltage-dependent sodium channels. So if you don't find a good effect with optogenetic stimulation, it may be that the neurons are not sufficiently stimulated. Okay, there has been confusion in the literature about that. All right, so we see that this dopamine stimulation generates a choice bias. That's compatible with the idea that the animals seek this dopamine stimulation that comes not only from the reward, but also from the artificial stimulation, right? Let's see if the neurons do that also. So we stimulate optogenetically, plus give the reward, and at that point we get a very nice response to this stimulation. Whereas when we give a drop of liquid without this optogenetic stimulation, we get a decent response, but it's smaller, okay? And then when we do that repeatedly, and we check the response to these two stimuli, the blue and the red stimulus that we saw that produce a choice bias or choice difference, we see that the response to these stimuli is higher when that stimulus has been followed by the reward plus the optogenetic stimulation compared to the stimulus that's only followed by the same reward, right? So the neurons also recognize this artificially enhanced value through the optogenetic stimulation. That's of course a reinforcement effect. That means the neurons were stimulated at the time of the reward, much later, beyond here, and the response to the stimulus is enhanced, okay? The agents, humans, animals seek dopamine excitation. They actually don't seek the reward for its own sake. They seek reward as a means of getting dopamine excitation. All right, here. So this is from Krakow. You get all these beer drinkers, and they probably eat something. These are your friends. So you're seeking liquid, food, and social rewards when you go to the pub, right? Maybe a little TV too. Well, I'm not sure. What you do is actually, you do this, you seek these foods and social rewards and liquid in order to get the dopamine excitation that they elicit. You're actually going for the dopamine excitation. You want the dopamine excitation, and how do you get it? Well, by going to the pub and having a beer and having food, right? I think I'm going to stop here. Thank you very much.
Thank you, Wolfram, for this wonderful lecture. Very stimulating, and I guess we're already salivating over that grilled steak that may be coming our way. But before that, there's chance for a few questions to clarify any remaining unclarities.
So I have a question about this effect where you have unexpected reward, and before that you have a stimuli. So there is a dopamine signal, dopamine activity after unexpected reward. But then it somehow moves after the stimuli, right? You have higher activity of dopamine after the stimuli. So my question is, what is the or some assumption, what is the circuit inside the brain which produces this activity that dopamine moves back, like somehow back when you repeated the same sequence?
Yeah, that's a real problem. There are 35 inputs to dopamine neurons. None of these inputs show the same response that the dopamine neurons do. So you're asking for two things. You're asking how does the dopamine response transfer backward in time, basically, to the earlier stimulus in the next trial. What is the input at all for the prediction error response? There is a thing called the eligibility trace that indicates that when a neuron, like in the striatum, is active, it carries an enhanced calcium signal or something for a few seconds, and onto that signal, the dopamine neurons, that's a pure theory. There are a few experimental indications, but it's by no means clear. The dopamine signal then acts on this eligibility trace and modifies the synapses between the input to that neuron. Well, the input of the neuron and the neuron itself. Yes. So you have an input to the striatal neuron, you have the striatal neuron, and there's a synapse, and then the dopamine neurons come in and modifies that synapse. And you can only modify that synapse because the reward signal comes 2 seconds or whatever later. You can only modify that if this synapse is modifiable, called eligible for modification. That's the idea. So that's the local circuit. There's a nice anatomical paper, several papers from the Oxford group, including Paul Bolam in monkey. Which brain structures really are involved in that, I don't know. As I said, there are 35 inputs to dopamine neurons. None of them have the same signal.
But at the end, probably somehow stimulation from sensory signal should stimulate then, as you said, through striatum somehow dopamine, but then that needs to move back in time. I mean, some other sensory dopamine needs to stimulate some other sensory signal somehow to go back, and then there should be somehow a circle, right?
Yeah,
somehow.
That's the point. That this sensory stimulus occurs, the input from the cortex that generates in this, let's say, striatum, the sensory stimulus, the sensory response in the striatum, that needs to be enhanced by a later signal. Right, so there must be some memory that this synapse has just been used, because you cannot just, the dopamine signal cannot enhance every synapse. Okay, but I only answered your question half, because what you were actually asking is how the dopamine neurons themselves learned that. I only talked about the striatum. Well, that makes it even more complicated. That means you have to go from the striatum probably to the dopamine neurons. There's a connection through a GABAergic pathway to the pars reticulata of substantia nigra. These are GABA neurons that synapse on dopamine neurons. It's double inhibitory circuit that people know you can stimulate in the striatum and you get an excitation in dopamine neurons. That's a possibility, yeah. Okay, so the circuitry is, as you see nowadays, since 5 or so years, everybody works on circuitry. It gets complicated.
Other questions on Zoom, and then we would move on.
I have a question if all of those effects were universal to all of the dopamine neurons in the VTA, or is there a possibility for some like cell type specificity?
I wouldn't call it specific, but there are some neurons that don't respond. Even in the VTA, in the substantia nigra, pars compacta are more neurons that don't respond to reward. That means in the VTA about 95% show this reward prediction error. In the substantia nigra it's anywhere between, the boundary is even not 100% clear, but it goes down all the way to 55-60% of neurons respond, the others don't do anything. Plus, the individual neurons, there has been a paper by Uchida and Botvinnik in Nature maybe 2 years ago. The individual neurons have a different sensitivity to positive and negative prediction errors. Some neurons only code the positive prediction error and basically nothing for the negative prediction error. These are the so-called optimistic neurons, and the other extreme are neurons that code very steep the negative prediction error and don't care much for the positive, and there are neurons in between. And when you take the whole population, you get a nice prediction error signal. But we've seen that. We've seen these variations before. It's very obvious.
Another question, is there a way to try to distinguish those neurons?
Sorry, to do what?
To distinguish those neurons that won't respond or
I bet you. I think so. There must be some molecular difference between the neurons. I'm not sure that this is a category A, category B, category C, but I would think if one looks very carefully at the genes in these neurons, one might find some difference. Yeah, and then here you go. Then you stimulate these neurons and see what happens. You stimulate these neurons, and then you have the whole thing working for you. Yeah, that's nice. Yeah.
All right, we have time for like 2 more questions. We'll have some here.
Hey, so from the beginning of the presentation, when you were talking about picking one beer, I was starting to think, what about habituation? Is it something that you're investigated? And if yes or no, would you expect the habituation to occur within the dopaminergic system or within the sensory system, which will interfere with the propagation towards the
I didn't get the beginning of your question. About
habituation, the effect of habituation on
of what?
habituation. So
Habituation.
yes, like excessive exposure.
I'm not sure, but dopamine responses don't habituate.
They don't habituate.
Yeah,
So
yeah. You get them after you get them in 20 trials. The 20th trial response is the same as the first response. They don't habituate.
not even like habituation over the sensory activity for the specific stimulus, which
No. If you have a new stimulus and you have a reward, then they respond to the reward, and after a while they respond to the stimulus and they lose the response to the reward. That could be called habituation, but that's not a habituation. That is in fact the increasing predictability of that reward that reduces the prediction error response. But no, these neurons don't habituate. Yeah.
okay, sure.
I guess one could ask, does the behavior habituate if you keep doing this with monkeys, or do...
No, they satiate. The behavior satiates, the animal satiates, if that's what you mean, that's interesting, yes. The animals satiate, and we have tested, we have compared neuronal responses we record like 3-4 hours, usually in the morning. And we compared responses in the first hour and the last hour of these recordings. Maybe this is what you mean. Then the responses stay the same, although the animal stops working just after we record the last. So we have 4 hours, we record the first hour, we record the last hour, and just after our recording, the animal stops working. In this case, the animal usually works quite okay, and then suddenly they say, this is enough. They do a couple more movements, and that's it. So the animals have quite an all or none response to their cooperation. But we have another experiment, totally different in orbital frontal cortex, where we see that the satiety actually continues gradually, goes gradually, but the animal only stops at one point. Mm-hmm. In this case, the dopamine neurons do not change their activity. And the answer is that they adapt to the reward value. If you have a small and a large reward value, then the smallest reward value produces the smallest response, and the largest reward value produces the largest response. If you reduce the value distance from a small reward to a somewhat intermediate reward, the neurons still respond to that, and not the other reward. The neurons respond to the intermediate reward maximum, just like to the big reward. If it is clear in a situation that, like the first hour and the last hour, we saw no difference in the responses between the first and the last hour, even though the animals stopped working 10 minutes later, right? If you mean that by habituation, then yes, the neurons adapt their activity. The response does not go down. This is called adaptive coding. That's known in the visual system as well. We adapt to the so-called reward probability distribution. In this case, the variance and the mean are reduced, but the response stays the same to the maximum reward, as if the slope of a response becomes steeper. We've seen that in the orbital frontal cortex systematically, and we have published a paper in 2005 on the adaptation. It's not as clean as I explained it, because we discovered that after the experiment.
Okay, thank you.
Okay, so no habituation and no satiety response, probably because of adaptation. Yeah. Okay.
Thanks so much for the lecture. I was wondering, have you seen examples of where the prediction error has gone wrong. So for example, I would imagine in schizophrenia, obviously the dopamine hypothesis is still a bit debated. Would the prediction error be, go wiry in schizophrenic patients, or would you know of any other diseases that...
Where the prediction error response went wrong?
Yeah, so sort of how neurons don't respond in the same way, for example, with schizophrenic patients.
You can easily get wrong responses when the animal doesn't pay attention. Yes. In particular, later when the animal gets sated. Yes. You can also, if you time the stimuli too close to each other, the response is reduced. We called it overtraining. We had almost a paper on that, until we found out that probably when you do repeated trials with a short interval, then everything is predicted by all the stimuli. So they respond to the first stimulus and nothing else. Yes.
So
but also the neurons may not respond if the animal is not looking at the stimulus. We've seen that. People sit there, this is the third neuron today that doesn't respond. And I say, well, have you checked the eyes? Oh no, the camera is broken. Okay. But the animals usually fixate, but towards the end, when they get sated or bored or something, then monkeys are capricious animals. They can fall asleep in the chair, like we do.
Yeah, so essentially, when prediction errors don't work, would it be usually an attentional issue and not something like there wouldn't be a biological deficit that leads to prediction errors not working?
I don't know. I would think, you always want to relate a neuronal response to something in the outside world. To assume that the animal is dreaming and still looks at the stimulus but actually dreams, this is very difficult to do. But when you see the animal is drifting off with the eyes and the responses go down or hardly there, then yes, then you have something. That means what I'm saying, we can only use positive evidence for a behavioral alteration, right? Whether we see in the responses that in 20 trials there are sometimes just no responses. 1 or 2 trials there's no response. That happens, we don't know why. That is the only thing I know. Yeah. That is uncontrolled.
The main reason I was thinking of schizophrenia is because sometimes they will have reward signals to things that shouldn't be associated with reward, and also the other way around. So if you something that's supposed to have a positive reward, they just don't respond to it whatsoever. And so I was just wondering whether that would be related, but I guess maybe not.
I think there might be difficulty with having schizophrenic monkeys to record dopamine neurons from, and that the patients typically object having their dopamine recordings done in their brains. So there may just not be any data to what happens in schizophrenic patients with dopamine neurons.
No, the only human dopamine recordings are from Parkinson patients. And that is, I have not done that myself. I know some data, I'm not sure, I cannot comment very much about it. But Parkinson and schizophrenia is pretty much the opposite picture in terms of dopamine function. So that may not happen in schizophrenic patients, yes. Schizophrenic monkeys, I have no idea if there's such a thing. Probably is, but yeah.
Right, we have one more question there.
Hi, thank you so much for your presentation. I just have a quick question. Whether there is a change for the same animal in terms of response, if you consider the effect of aging of the animal itself, or
aging?
Yes, and the movement.
I don't know. Our monkeys get pretty old. We start recording them at the age of 4, and we record for 2 or 3 years. But at the age of 8 or 10 years, a monkey is like a human in their 40s or 50s or so. They don't get that old. But then the monkeys can get 16, 18 years old in a natural life. That happens. I don't think we had any monkey that died because of age. No.
No, my question was considering the nigrostriatal compartment and whether the nigrostriatal compartment and whether there would be a change for the recorded response to the stimulus.
I don't
Consider-
understand.
The question is whether if you have recordings in aging monkeys, which I guess you just said there aren't so many in really aging monkeys, whether the dopamine responses would change with age, like they might be in pre-Parkinson's patients.
No,
something like that.
I don't know. Humans lose about 30 to 40, maybe 50% of dopamine neurons between the age of 20 and 60 or 70. But I would suspect that the other dopamine neurons probably would still respond. And those that have disappeared, you'll never record from. Yeah, I can only speculate. Yeah, okay. Yeah, sure.
We have time for actually a few more questions. Turns out the food waits for us only at 6:30.
Okay, thank you. I will try to be short. Thank you for the wonderful talk. I have a question about the individual differences, because
About what?
Individual differences. Because when we think about maximizing value of a reward, it is also a question about impulsivity. So we know
Sure.
that humans and probably
Temporal
also
discounting.
animals differ in this regard. Do you think that this difference lies in dopaminergic neurons? Or it's the same question about like risk-taking?
The risk-taking is an easy answer. They are pretty much all risk seekers with low rewards and become risk neutral, and risk avoiders with large rewards, larger rewards. What was the first part?
You talk about maximizing the reward, and then if it requires some planning, yeah. So
Sure.
the choice depends on like impulsivity.
Oh, yeah.
The
temporal
difference between
discounting. Yeah.
immediate and delayed response.
We only used 2 or 3 monkeys in the study, and I think the temporal discounting study was probably only on 2 monkeys. And we published, I just looked at it today, at the paper 2008, and they have pretty similar discounting functions. But I have to say, I once presented behavioral data from 4 monkeys about some social thing, and people killed me that it was not enough monkeys to say something, so I'm not going to say that again.
Okay, thank you.
But yeah, for the risk, they are very similar. We have plenty of, maybe easily 10 monkeys for different experiments tested in risk. The temporal discounting experiment was only in 2 monkeys. You could call risk attitude also impulsivity. Yeah, but then in that case, they were, if you take risk attitude for impulsivity, then they were pretty similar. Yes. Yeah. Okay.
Any questions from Zoom, I guess? No? And then I think, thank you so much. We'll want to thank Dr. Schultz again. I guess Daniel can have us further guidance.
Yeah, I hope there will be some guides here, but essentially the barbecue is in the other department, which you guys know. It's on the patio. Magda, do you know will you take the people or will some of our volunteers? Okay, so thank you.